Hi everyone, Organic Genetics Tutor here. So today I'm going to be going over some Unit 2 material. I didn't make a video over this because I anticipated you guys would do so well on Exam 2, which you guys did. I'm very proud of you guys. So for Unit 2 you guys went over a variety of topics, of them being heritability. So heritability is essentially just a measure for how well a trait can be inherited. There's two types of heritability. There's broad sense heritability and narrow sense heritability. Broad sense heritability, or big H2, essentially asks, how does the genome affect phenotype? So what that means is, broad sense heritability essentially measures the total genetic variation in a trait. And that equation is big H2 over VG over VP, where VG is genetic variation and VP is phenotypic variation. Narrow sense heritability, also known as little h2, essentially asks, how does one gene affect the phenotype? Now what that means is, narrow sense heritability only, or essentially just measures the additive genetic variation, or just the variation that one gene can have on the phenotype. And that equation is little h2 over VA over VP, with VA being additive variation and VP being phenotypic variation. If an individual has higher narrow sense heritability, or H2, that means that that individual has a better response to selection. To have a better response to selection, that means that this individual has a greater response to the environment, to changes in the environment. So the phenotypic variation that occurs in this individual is, if it's high, it's dependent on the way that the environment is. If the H2, or narrow sense heritability, is low, that means that mainly genetic variation is what's causing differences in the phenotype of the individual, regardless of the environment. So higher H2 means that the environment plays a bigger role in the phenotype. Lower H2 means that the environment does not play a large role, rather genetics plays a larger role in the phenotype of this individual. So here's a question. A peach breeder has an orchard of peach trees with an average fruit mass of 30 grams. The breeder selects the top 10% of plants to reproduce for the next generation. The selected plants have an average fruit mass of 45 grams. The next generation has an average fruit mass of 36 grams. What is the narrow sense heritability for fruit weight in this orchard? So for this problem, we are not directly given VA over or VP, so we are going to use the equation little h2, or narrow sense heritability, equals m2 minus m over m1 minus m, which you have m, which equals the original mean, m1, which equals the selected mean, and m2, which is the progeny's mean after the selection has occurred. In this case, we know that a peach breeder has an orchard of peach trees with an average fruit mass of 30 grams. So the original mean, or m, in this case, is 30 grams. The breeder selects the top 10% of plants to reproduce for the next generation. The selected plants have an average fruit mass of 45 grams. The selected mean is 45 grams, so m1 equals 45. The next generation has an average fruit mass of 36 grams. So the weight of the progeny after the selection has occurred is 36 grams. So that means that M2, or the progeny mean after selection, is 36. Now, all we'd have to do is simply plug this into the equation. So, narrow sense heritability equals M2, which is 36, minus M, which is 30, over M1, which is 45, minus M, which is 30. That equals 6 over 15. Simplify this down even more, that equals 0.4. So that tells us the narrow sense heritability in this, I guess, progeny of the plants, or for this organism of plants. Now, if we were given another plant, and we found out that the narrow sense heritability was 0.3, and we were supposed to compare between both of these, which responds better to selection. Here we see that this is 0.4. So this is a higher narrow sense heritability as compared to 0.3, which means that this right here would have higher selection or better response to selection, meaning that the environment will cause changes in the phenotype to occur more readily as compared to this individual with that, or that species of plant with that H2 or narrow sense heritability. Going on to one of the more major topics you guys spoke about, and that is the topic of 3-point crosses. All right, so for this problem, we have cookies as our example. So in snickerdoodles, the cinnamon, sugar, and egg loci are linked with recessive mutations C, N, S, and EG respectively. You cross a homozygous wild-type snickerdoodle with a snickerdoodle that has all 3 mutant phenotypes. You then test cross the progeny. The results of the test cross are shown below. Construct a genetic map for these 3 loci based on your results. Then determine the coefficient of coincidence and interference for these loci. All right, so the way I like to go to solving these problems, I like to follow three steps. The first step is to find the parentals and double crossovers. I'm going to simplify double crossovers as DXO. So parentals are going to be the 2 highest numbers that occur in the progeny, whereas double crossovers are going to be the 2 lowest numbers that occur in the progeny. If we look at all the progeny, we see that we have 54 and 65 as the 2 highest numbers. We have 3 and 4 as the 2 lowest numbers. So we know that the parentals are going to have the genotype CNSEG, though we do not know the particular order, at least not yet. And we know that the double crossovers have the genotype CNS plus, as well as plus EG. So now we have identified the parentals and the double crossovers. What are we going to do next? Now we want to find the gene in the middle. What is the middle gene? Now what does that mean? What significance does it have? Here's just a small example. If we were to have an individual who had 2 chromosomes, one of them having all dominant genes, ABC, and the other having all recessive, A, little b, little c. If you were to have a crossover occurring here between genes A and A, that would result in us having an individual, or having the chromosomes being little a, big B, big C, big A, little b, little c. Now if we were to have another crossover occur between C and C, that would ultimately result in us having chromosomes that have little a, big B, little c as one of the genotypes, and big A, little b, big C as the other genotype. Now if we look at the original genotype and we compare between the original genotype and the final genotype, it appears that B, the gene in the middle, is the gene that flipped. Whereas we know that A flipped with A, and little c flipped with the position of big C, and big C likewise flipped with the little position of little c, and that resulted in us having this genotype. So for double crossovers, we want to find the gene in the middle, because that tells us that this gene in the middle is the gene that did not switch, and the two other genes are in fact the genes that did switch, which comes in the name, double crossover. Here we had one crossover, here we had another crossover. So the key to finding the middle gene is to look at the gene that appears to have changed. So if we were to compare between this and this, it looks like B flipped when we know that B did not flip at all. So that tells us that B is in the middle. It's kind of confusing, but we're going to continue through this and hopefully it'll make more sense. All right, now let's write out both the genotypes. We're going to write out the parentals. CN, S, EG, plus, plus, plus. And now we're going to compare these with the double crossovers, which is CN, S, plus, and plus, plus, EG. So, comparing between both of these, we can see that CN and S appear to have stayed in the same position, and EG is the gene that appeared to have flipped between both of these. It appears that there was a flip right here. However, we know that the middle gene is the gene that looks like it flipped, but in reality, it didn't flip. So this tells us that instead of having a crossover between EG, there was actually two crossovers that occurred between CN and the corresponding plus gene for CS, and S and the corresponding wild type allele for snickerdoodle. So that tells us that EG is in the middle. So now what we're going to do is we're going to rewrite this in the genotype. Rewrite this genotype with EG being in the middle. So the genotype appears to be something like CN, EG, S, with the pluses all corresponding for each of the specific genes. So now that we know which gene is in the middle, the second step is to compare the single crossovers. And I'm going to simplify single crossovers as SXO. Here's a picture of all the progeny. We know that these right here are the parentals, and these right here are the double crossovers. That means that everything else is going to be the single crossovers. Now simply what you want to do is you want to find out which crossover occurred in order to get one of these single crossovers. If it was a crossover between CN and plus or S and plus. That's what we're trying to find out now. So what we're going to do is we're going to compare that with the parentals. So to compare this, let's start off with this crossover, the single crossover. In order for us to get CN plus and plus as a genotype, we have to have a crossover occur between CN and plus. So there has to be a crossover in which CN goes down to where the plus is, and the plus crosses over with the CN, and that will give us this genotype. So that means that this right here is a crossover occurring between CN and EG. On to the next chromosome. We're going to do plus SEG. In order to get this genotype, we have to have a crossover occur between plus and Cn. So there has to be a crossover occurring here. So this, likewise, is a crossover between Cn and Eg. And what we're going to do is we're essentially just going to keep this up until we get through all the single crossovers. The next chromosome we're going to do is this individual right here, with the plus S plus genotype. Now, I forgot to mention this. On this problem above, the easiest thing for you to do is when you rewrite the single crossovers, write the middle gene in the middle and the corresponding genes in the positions of the other corresponding genes. In this case, it didn't matter so much because these are both pluses. However, I just now realized in the problem I did above, for this individual, the plus SEG, the way you'd want to write it out is you want the middle gene being in the middle, such as EG right here, and you'd want S being in the position corresponding with S. It just makes it easier to see what gene might have flipped. So for this gene, right here, the plus S plus, we know that, at least from the way we're writing it, S is on the end, and plus, or EG, is in the middle. In order to get this genotype, you have to have a crossover occur between EG and S. So this S crosses over and switches places with the plus, and the plus switches places with the S, and that gives us this genotype. So we know that this is a crossover between S and EG. Onto the chromosome underneath it, CN plus EG. Remember to write it out in the proper format. CN EG plus. In order to get this genotype, you have to have this plus switching with this S, and this S switching with this plus. So it's a crossover occurring between EG and S. Now we have identified which crossovers are associated with, or at least which of these chromosomes or genotypic expressions are associated with each crossover. Now what we want to do is we want to calculate the recombination frequency for each of these crossovers. So we want to calculate the recombination frequency between crossover of CN and EG and crossovers of S and EG. So for a crossover between CN and EG, the way to calculate this is you want to put the single crossover and you want to add it with the other single crossover for CN and EG, and you want to also add that with the double crossovers. So you're adding in all the crossovers that have occurred between CN and EG. And the reason we put the double crossovers is because the double crossovers, as we've been shown from above, occurs between this gene, but the only difference is it occurs between the other gene. But it still occurs between CN and EG. So we're still going to include that. Likewise, when we're calculating the recombination frequency for the other crossover, like S and EG, we're also going to include the double crossovers. And then we're going to put all of this right here over the total. So if we look at the progeny for the single crossovers, we know that these two crossovers right here were between CN and EG, because we calculated them right here, and we found out right here. And the values are 30 and 27. So we're going to have single crossover, 30 plus the other crossover, 27 plus both double crossovers, which were 3 and 4, over the total. Total is, if we add this all together, I'm great at math, so that's 211. if we plug that in, that will give us 0.303 or 30.3% recombination frequency. Now we're going to do the same for the other crossover, S and EG. For S and EG, we had 16 and 12. So the single crossover is 16 plus the other single crossover, 12, plus the double crossovers, 3 plus 4, over 211. That gives us 0.166 or 16.6% recombination frequency. What does this mean? Okay, we know that this tells us that the frequency of these crossovers occurring is this percentage, but what significance does that have in telling us how far these are apart on a genetic map or on the chromosome? Well, for every single percent we have of recombination frequency, that is directly equal to 1 map unit, or 1 centimorgan. Map unit, centimorgan, same difference. It doesn't really matter what you choose to use. In this case, I'm just going to stick with map unit, but it's also known as centimorgan. So if we have 1% recombination frequency, that means that this is 1 map unit apart from the desired gene the desired 2 genes we're looking at. If it is 20% recombination frequency, that means that this is 20 centimorgans or 20 map units away from the desired gene that we're looking at. So if we had A and B, that would mean that this is, if we have a 1% recombination frequency, this is 1 map unit apart from one another. If we had 20% recombination frequency, that would mean that these right here are 20 map units apart from one another. So now that we have the recombination frequencies, we're able to construct what the genetic map might look like. We know that we have CN here, we have EG in the middle, and here we have S. For CN and EG, that is 30.3 as a recombination frequency, which means that this is 30.3 map units apart. For EG and S, we know that the recombination frequency is 16.6, or these right here are 16.6 map units apart. And that gives us a total of 46.9 map units between all of these genes. So now we found step 2. Now let's go to step 3, which is to calculate the interference and coincidence and coefficient of coincidence. In order to calculate interference, you have to use the equation I equals 1 minus the double crossovers we had over the expected double crossovers in order to find the expected double crossovers, we simply have to take the frequency between both of these crossovers and multiply them by each other. In this case, CN and EG is 30.3 map units, or 30.3 frequency. But if you were to write that down as a percentage recombination frequency, in a frequency, it's 0.303 times 0.166, which is just the original numbers that we got here, and that equals 0.0503 or 5.03%. So that means that we would expect 5.03% of the population to have a double crossover in the genes. Now, in order to find the total amount of individuals who are going to have a double crossover, we just take this percentage, or we take this frequency right here, and we're going to multiply this frequency right here, 0.0503, with the total population that we have. The total population of the progeny is 211, and that gives us a total of 10.6 individuals we would expect who have a double crossover. Now we simply have to plug this back into the equation of I equals 1 minus the double crossovers. We know that we had 2 double crossovers here. The total of these double crossovers is 7. Oof. 1 minus 7 over 10.6 equals 0.3397, which means that 33% of double crossovers will not occur. So when a crossover occurs, you have these chromosomes, which then become all, coiled around one another. So it makes it more difficult for another crossover to occur due to these chromosomes becoming all entangled with one another. So when you have one crossover, the probability of another crossover occurring becomes lower. And this is just telling us the percentage of double crossovers that should have happened that didn't occur. So 33% of these double crossovers that were supposed to occur did not occur. And that tells us the interference, or the rate of interference. All right, going on to one of the other topics you guys spoke about, which is X linkage. So genes located on the X chromosome is what's meant by X linkage. With X linkage, you have X inactivation may occur when a chromosome is inactivated and becomes a bar body. So when you have 2 X chromosomes, in order to ensure that you're not overexpressing this chromosome, you have one of these chromosomes that turns into a bar body. So essentially it becomes deactivated and then it crumbles up into a ball-like structure, which is then known as a bar body, and it's not expressed. And this happens randomly between X chromosomes in different cells. For this problem, we have a boy with Klinefelter syndrome, and he has 47 chromosomes with an extra X chromosome. And he is born to a mother who is phenotypically normal, and a father who has the X-linked skin condition called anhydrotic ectodermal dysplasia. The mother's skin is completely normal with no signs of skin abnormality. In contrast, her son has patches of normal skin and patches of abnormal skin. A. Which parent contributed to the abnormal gamete? So for A, we know that the mother is normal, and we know that the father is normal. Or before I go into this, let's say that the chromosome, or the X chromosome that has the condition is XC. And that results in the skin condition. Here we have 2 parents. We have a mother who is phenotypically normal, so we're going to assume that she's also genotypically normal. And we have a father who has this X-linked skin condition. Coming together, they will then result in having an offspring who has the genotype XCXY. So, to answer part A, we can assume based off of the skin condition that the father has that he is most likely the individual who is to pass on this aneuploid gamete. or this aneuploid or abnormal X chromosome. So now we know that the father is the person who passed down this abnormal X chromosome. So for part B, use the appropriate genetic terminology to describe where the mitotic mistake that had occurred. Be sure to indicate which division the mistake occurred. So now we want to see where the non-disjunction occurred. So we know that the father has a genotype of XCY. So there's two different possibilities that this could have occurred. This could have occurred in meiosis 1, and this could have also occurred in meiosis 2. Had we had a non-disjunction occur in meiosis 1, let's just draw it out. Here we have meiosis occurring. We have a duplicated X chromosome, and here we have a duplicated Y chromosome. If this non-disjunction happened in meiosis 1, a non-disjunction means that the chromosome went to the wrong side of the cell. So let's say we had non-disjunction occur here in meiosis 1. That would mean that this entire chromosome would then go to the side that the Y is on, or the Y goes to the side that the X is on. That would result in us having one chromosome that has no genetic information, or one cell that has no genetic information, and one cell that still contains both of these duplicated gametes, or these duplicated chromosomes. If this was then to proceed in meiosis 2, that would then result in us having two cells containing none of these chromosomes, and these two cells containing both of these chromosomes. If this were to happen in meiosis 2, then we would start off meiosis in the same way we started off the previous meiosis 1 problem, with a duplicated X chromosome and a duplicated Y chromosome. We would have meiosis 1 occur normally. So then we would result in us having a duplicated X chromosome in one cell and a duplicated Y chromosome in the other cell. But since the non-destruction happened in meiosis 2, that would mean that instead of this sister chromatid going to the opposing sides of the cell, one of them is going to go to the wrong side, resulting in us having two cells, one of which containing none of the genetic material and the other one containing both X chromosomes. However, for this other cell, we're going to assume that a non-destruction did not occur unless we have some sort of strange individual with all these weird chromosomes. Now keep in mind that this also can happen for this Y chromosome as well. So we could have a regular X splitting pattern for the X chromosome, and we could have this non-destruction occur for this Y chromosome. It just differs based off of what extra chromosome the individual has. All right, so we have both of these genotypes. We want to figure out which meiosis event or which non-disjunction event could have ultimately led to this individual having the genotype XCXY. We know that the mother is just going to pass on an X chromosome. So the mother, she's just going to have a cell with an X. So I should probably just draw it like this. If we were to include it with this individual, or this cell right here, we would then have an individual who has 3 X chromosomes. We know that this individual is a boy, meaning that he has a Y chromosome. So the only other possibility would be if we had this cell right here contributing to the non-disjunction event, or fusing with this other female gamete, resulting in us having XC, X, and Y, which gives us the genotype of the boy. So this non-disjunction event occurred in meiosis 1. Part C, using the appropriate genetic terminology, explain the son's skin phenotype. So the son, he has patches of normal skin and patches of abnormal skin. That is indicative of X inactivation. So this boy has random cells in his body. He has, let's draw two cells. Here we have XC, X, and Y. XC, X, Y. In this cell right here, we have this XC being inactivated. So all the cells that are going to be produced from this cell via mitosis are going to have a working X chromosome. So these right here are all going to produce normal skin. However, in this cell, here we have the normal X chromosome that is turned into a bar body. And recall, X inactivation occurs randomly between X chromosomes. So this X chromosome is randomly going to be turned off. So all the cells that are produced from this cell via mitosis are going to have abnormal skin. It's too small for me to write it. You guys get the memo? Yeah, so that is in X inactivation.